Hello everyone, welcome back to another session of RNA biology. So we were here in the previous class, we were learning the importance of vitamins and omega-3, such kind of molecules in real time affecting the length of telomeres. And we believe that a lot of such molecules based on the data in available from the population and also in experimental models, we have shown that the telomere length remain quite healthy if the people are taking healthy diet and continue to be living in less stressful environment. And longer the telomere length, the less likely the cell will undergo apoptosis or cellular senescence. So that is the take home message. Now let us think about some telomerase activators. So telomerase is an enzyme which extends the length of telomeres, and we know somatic cells do not have telomerase activity, even if it is there, very marginal. And we also saw some non-coding RNA regulating the telomerase function in the previous classes. So we should keep in mind that there is still scope for enhancing the telomerase activity, not to a level that it can cause cancer, etc. So don't assume that activating telomerase is leading to cancer. The answer is no. If that is the case, stem cells and germ cells should not have a telomerase activity. So telomerase activity do not cause cancer, but high telomerase activity is a necessity for the cancers to survive. So telomerase activity do not cause cancer, rather it is a boon. But evolutionarily, our somatic cells do not have telomerase activity as it has in the stem cells as well as in the germ cells. So can we think of activating the telomerase? So activation of telomerase has been shown to reverse the aging process in the cells, tissues and even in the whole organisms. So let us think more about it. Telomerase can immortalize the cells, and we have seen that one in around 10 million cells can gain this telomerase activity, and that will lead its way into immortalization. So we know that. So a study that is published in the journal Science have shown that human retinal pigment epithelial cells and the foreskin fibroblasts were transfected with vectors encoding human telomerase catalytic subunit HTERT, and the control cells showed the telomerase shortening as expected, whereas the marker of cellular aging, that is the beta galactosidase, is found to be at normal levels in control cells. However, when you express the telomerase, the transfected cells exhibited longer telomeres and reduced the levels of beta galactosidase. So what it indicates, cellular senescence as reflected by the marker, the beta galactosidase itself is reflected. That means the cell is not planning to undergo senescence because of the presence of telomerase. By the time the study was published, the telomerase expressing cells had exceeded their expected lifespan, which is more than 20 generations. Means what is the maximum number of generation that cell can live in a cultured condition? Beyond that, it went to 20 more generations. So this is a interesting supportive data to show that telomerase or the activators of telomerase can reduce the aging of the cell. So telomerase can restore the youthful phenotypes in live tissue. So normal human dermal fibroblasts were transfected with HTERT and then grafted onto mouse skin, on the skin of the mouse, and where you have reprogrammed this HTERT, means overexpression of the HTERT, which is the telomerase catalytic component. So mice grafted with the telomerase negative control cells exhibited phenotypic signs of cellular senescence, that is increased fragility, reduced levels of collagen 1 and 3, and subepidermal blistering, that is sign of old age. So what happens? Mice grafted with the telomerase expressing, that is HTERT expressing cells, exhibited quite very youthful phenotype, and despite the same number of replications. So it's not that telomerase expressing cell is not dividing or something, it is happening, dividing everything as usual as a control cell. However, being a proper telomere length available in this HTERT expressing cell, they did not exhibit the senescent cellular senescence. So these data suggest and support the view that telomerase can be reactivated for an extended tissue life or cellular life. And the telomerase activation can restore the youthful phenotypes in the whole organism itself. A study conducted in 2010 showed, that was published in the Nature, the journal Nature, showed that the telomerase activation in mice dramatically reversed the effects related to aging. So, a restoration of youthful phenotype was seen in the organs such as testis, spleen, liver, and intestine. These organs are used mainly because they have a accelerated rate of cell replication or cell division. So, additional effects of telomerase activation included restoration of fertility, reversal of cerebral atrophy, reactivation of neural progenitor cells. So, these positive effects also contribute to keep the brain young, or the performance of the brain quite active as it happened during the young age, and fertility, because anyway the germ cells have the telomerase activity. However, the starting cells of the organ testis also have the telomerase activity, make them in a much pristine condition as it occurs in the young age. So, telomerase activity also can be an indicator of the longevity. So, several experimental studies have shown that multi-generational study by Ashkenazi Jews, it's a community of Jews. You can read a lot about Ashkenazi Jews. They also have also a lot of consanguineous marriages and consanguinity related stories. Also, you can hear a lot about Ashkenazi Jews, and they have been used a lot. Used means they have been, their data has been used extensively in genetic studies. No humans can be used in a genetic study, but the data can be used for study. That has happened with Ashkenazi Jews a lot. So they have been known to have a exceptional longevity. So a parent group of around 86 members, and they have an average lifespan of 96 years, and the offspring group of around 175 members, and the control group were around 93 members. They found that the population exhibited abnormally high telomerase activity mediated by a mutation in the HTERT, which is the catalytic subunit of the telomerase activity. So, because of this mutation in Ashkenazi Jews, they find that the telomerase is performing more than usual. So, high telomerase activity, credit goes to the mutation, and because of this, when the telomerase is functional, they have a longer telomere than usual. So that's what, say, a normal person have, like we saw, that around 11 kb is the average length. But in their germ cells, in their somatic, like in stem cells, the telomerase activity is so high, it can exceed the 11 kb. So that even if the cells are dividing at a normal rate, it will never reach that so-called 4 kb length, which is a shorter telomerase length, because it is gone way too high. It is just like your starting money is only 1000 rupee, it can easily get exhausted. But if your starting is 1 crore rupee, it will take longer time to get exhausted, as long as the usage is more or less similar. The 1 crore is there, you spend in some extremely non-productive thing, like 1 crore, you put it in overnight, you can get rid of 1 crore. But if you are spending usually as the 1000 rupee fellow is spending, then the 1 crore rupee fellow have a lot of advantage, because to exhaust that 1 crore at the rate what a 1000 rupee holder is spending, then it will take longer time. So this is the concept you should have. the telomerase activity, when it is high, whenever it is functioning, it will add too much of telomere length. So that will take longer time to get into shorter length. So these results indicate the longevity of the telomerase activity can be connected because the average age of this parent group is 97 years, and probably the credit goes to the longer telomeres, and they do not show the signs of old age. And telomerase activators can be, there are many drugs also people have explored. So a naturally occurring highly purified single molecule derived from a Chinese herb known as astragalus, which is being marketed as TA65, and it activates human HTERT gene, that is the catalytic subunit. In vitro has shown that the moderately activated telomerase in human keratinocytes, fibroblasts, and immune cells. In vitro study means the petri dish studies, where you culture the cells and you treat with this drug, and you see the telomerase is hyperactive, which is reflected in the length of the telomeres. In vivo, that is inside the living animal, in vivo administered as a part of pattern protocol, that is a protocol for delivering these drugs, which the participants were given 10 to 50 milligram of TA65 per day for 12 months. And they have shown to exhibit a longer telomeres activity, and longer telomeres because of the activation of the telomerase activity. So many such herbal and natural occurring compounds have been tried. This doesn't mean that you can avoid getting old, but it can be delayed. Like many research is going on in expanding or extending the lifespan, or expanding the scope of individual cells in a body beyond their normal lifespan. So many such kind of research are going on. So let us think, what can you do for maintaining a healthy telomere and longer length of telomere due to the telomerase activity. So exercise is being shown to increase the telomerase activity. In murine model, they have shown that voluntary running for 3 weeks, and this exercise included around 2.9-fold increase in the aortic telomerase activity, because when you are running, your heart will pump more and you have more of blood supply in the aorta, so aortic telomerase activity is increasing, and around 3.3-fold increase in telomerase activity in the circulating mononuclear cells in the spleen. That is another observation. That is in mice. So in human model, compared to control groups, professional athletes exhibited a 2.5-fold increase in telomerase activity in young people, young athletes, and 1.8-fold increase in the telomerase activity in middle-aged athletes. So what you understand, that if you have a healthy lifestyle, means doing exercise and also leading a healthy diet and healthy lifestyle, that can have a increased telomerase activity, increased telomere length because of the good or effective telomerase activity, can reduce the rate of aging or can increase the life expectancy of an individual cell, and probably the aging can be delayed. Now we will move on to another topic, that is about the RNA modifications. We have seen a lot of RNA modifications, RNA editing, we have seen splicing, polyadenylation, etc. Now we are looking into some RNA modification which are not obligatory, that is a facultative. It can change temporally, it can change with age, it can change with a habitat or the food, the drug, what you take, those kind of things. So what you should keep in mind, this is something to do with the epitranscriptome. That is transcriptome, you have your genome is transcribed wherever the genes are to be expressed, and we call it as transcriptome. And the genes expression can be regulated by your dietary habit, your environmental habit, etc. We call it that branch of science as epigenetics, and we call the modification. We have seen some of them like H3K27 acetylation, H3K27 methylation, etc. These are all occurring onto the protein attached onto the DNA, and we call it as chromatin modifications. And the modification occurring onto the histone, we call it as histone code. And some modifications facilitate the gene expression, some modifications inhibit the gene expression. So we are still trying to understand the histone code in human. So it is not completely understood because a lot of dynamic behavior of this epigenome makes the study quite cumbersome. Many times these histone modifications we cannot link whether it is a causative or correlative. So causative link are hard to come by just because, like, say you got headache and that time one crow was sitting in the chair. So we cannot prove that the crow caused a bird sitting adjacent to you caused you headache. So you need to, that is a simple correlative. Next day also crow was sitting, you got headache. But even if it happens 10 times, we cannot establish that it is the causative, crow was the causative agent of your headache. It can 10 times also it can be correlative. That is the challenge we have in understanding the epigenome. So these RNA modifications that is occurring not as a requirement but as a possibility. So we call such kind of changes as epitranscriptomic changes, or we call these modifications as epitranscriptome. So one such important modification is N6-methyl adenosine. That is a modification of the N6-methyl adenosine, and we call it as M6A, and which is a abundant modifications in mRNA, and also it can be seen in the DNA, and it is a dynamic and reversible process. So we should keep in mind these modifications are reversible also, reversible process, and is most pervasive modification in human RNA. That means RNA of any given gene, there are so many A residues are there. In some selected residue, it can undergo modification and it can have huge impact in the rate of translation of that mRNA. If it is an mRNA, it can occur onto the non-mRNAs also. That means RNA that are not just mRNA, other RNAs also. It is found in starting from viruses, and most of the eukaryotes have it, including mammals, insects, plants, yeast, etc. Means whichever animal have got an RNA, it has the potential to have this modification, N6-methyl adenosine modification. It is also found in transfer RNA, ribosomal RNA, and small nuclear RNA, snRNA, as well as seen in several non-coding RNA, even such as X inactivation specific transcript. So the methylation of adenosine is directed by a large M6A methyltransferase complex containing METTL3 as the SAM binding subunit. S-adenosyl methionine is a molecule that is produced from your liver metabolism. So this molecule also have the ability, because it acts as a methyl donor. SAM acts as a methyl donor. So here the modification we are talking about is adding a methyl group. As you can see here, this is the adenosine, and all we have is a methyl group that is added onto the N6, nitrogen 6, and this methyl group CH3 is the methyl group that is added, and this modification need a methyl donor, and SAM acts as a S-adenosyl methionine is the methyl donor, and the modification occurs on the specific adenosine residues on the RNA. In vitro, this methyl transferase complex preferentially methylates RNA oligonucleotides containing GGACU. So this sequence is the GGACU. So this A can be the target, and a similar preference was identified in vivo also. In vitro means in petri dishes or in lab condition. In vivo means in living animal. Is mapped as M6A sites can be found in the Rao sarcoma virus genomic RNA, and also in bovine prolactin mRNA naturally to start with initially. So what we understand from here, RNA modifications such as N6 methyl adenosine, formation of N6 methyl adenosine with the help of methyl transferase enzyme. What is the enzyme? METTL3. So this enzyme can use the SAM as a donor to add the methyl group onto specific adenosine on various mRNAs. No matter whether it's mRNA, tRNA, doesn't matter. It can change the functional properties of that RNA, and many times it can be harmful. So N6 methyl adenosine is an abundant modification in the mRNA and DNA, and it is seen in various organisms as we speak, and they are present consistently across all the species we discussed. So M6A, that regulators can be divided broadly into three categories. Like in epigenome also, epigenetic modifiers also, you classify them into these three categories. So this epitranscriptome modifications such as M6A regulators can be also classified into three groups, that is writers, erasers and readers. Think in this way, that in a blackboard or in a green board, you are taking with a chalk piece and you are writing something. Let it be ABCD or poem or 1, 2, 3 something. So someone has to write it in order to have something on the board. Then someone should be able to read it, or someone should be able to erase it. So you can happen in any of this fashion. Like it starts with writing. Without writing, no one can read, no one can erase. If nothing is written, what are you going to erase? If nothing is written, what are you going to read? So writing is the initial step. Then it has to be read, or means it has to be sensed. Here, when you are reading, that image will form into your eye, and your brain will convert into signal, and your brain will process the meaning of that based on what training you got it. So this is called reading from a book or from a board. And erasing means you are just getting rid of that, what is written. So all these three molecules are present. That is why it is mentioned that it is reversible. If there is no erasers available, you can never reverse it. It is a permanent job. So it is writer is there, reader is there, eraser is there. So if the eraser is there, it is working opposite to the writer's job. So more recent studies have characterized other key components of the M6 methyltransferase complex in mammals, and this is basically the writers, means they create this modification in the adenosine residues, including METTL14 and also Wilmer tumor 1 associated protein WTAP and KIAA1429 and METTL5. So there are plenty and many more to come, because this is one of the recent discoveries in the RNA modification field. So you will find plenty more coming, because as I told you, like histone code itself remains enigmatic, although we know a lot, but we don't know completely. Like that, this RNA modification and the enzymes that cause this modification, such as writers, will be very difficult for us to comprehend in our more than 100 different types of tissue types, even in human model or human organism, one of the most important organism. The hypothesis that M6A in mRNA being dynamic and reversible is proven with the discovery of the first M6A demethylase. That means it is dynamic and it is reversible simply because they identified a demethylase, M6A demethylase, and this gene is fat mass and obesity associated protein known as FTO, and this is a classic example of eraser. That means writer adds the methyl group onto the adenosine and erasers gets rid of that. And there is one more eraser discovered, that is M6A demethylase, that is ALKB homolog 5, and is known as ALKBH5. So this was later discovered to be another important eraser in vivo in various cell types. So the biological functions of M6A are mediated through a group of RNA binding proteins that specifically recognize the methylated adenosine, and we call it as the reader. So here we are discussing about the writer, the eraser, and also the reader. And these binding proteins are named as M6A readers. So what we learned, that the RNA are vulnerable for modifications. RNA can bring in changes, and these changes can be of some crucial impact in the biology of that RNA. Means if it is an mRNA, its biology is linked strongly with the protein translation. And sometimes an unwanted protein or an overexpressing protein can be problematic depending upon which tissue and what protein you are talking about. So this is accelerated by writers and it is decelerated by erasers and it is sensed by the readers. So all these three molecules or group of molecules working together will bring in effective changes in the functioning of the RNA. We will learn more about this RNA modification epitranscriptomics in the next class. Thank you.